Hey guys, welcome back. Today we are going to do the digestive system. Let's pull it up and let's talk about it. So obviously Lab 10. As always, we're going to do some basic anatomy. I'm going to show you where the structures are on some labeled pictures. And then we'll go through, we'll look at our models. We'll see a little bit of histology. And then towards the end, I will talk a little bit about nutrition and recognizing certain macronutrients, and maybe even talk a little bit about what we need to survive. As always, you have an introduction. Feel free to read through it. It kind of talks about the basics of the digestive system. Um, but the main goal of this system is to take the food you consume and break it down. So all food comes in as big, hopefully whole food product, and it needs to get broken down to a very, very small chemical composition. So everything along this pathway is designed to break it down and absorb it, right? We have to break the big molecules into the small molecules, and then those small molecules get absorbed into the blood, so we can circulate these nutrients around and build new tissue and do awesome things that involve, I don't know, synthesis of protein, production of energy, you name it. So we'll start off where all digestion starts off, and that is in the mouth. Obviously, we have an oral cavity. We have teeth in here. We have muscles like the masseter that are involved in chewing and masticating food. So the food goes in there. We will talk about a variety of glands that are also located near the mouth. These are called salivary glands. You should know the parotid, the submandibular, and the sublingual. I will show you where they are. Obviously, the sublingual is located under the tongue, the submandibular is located by the mandible, and the parotid is located by your cheeks. These secrete saliva, and they contain various enzymes, and you want to lubricate the food so it could pass through the 30 some odd feet of digestive system that we have, and not get stuck. So some digestion does start taking place in the mouth, as well as a little bit absorption of carbohydrates and things. We have the pharynx, which is also part of the respiratory system. It's where the nose and the mouth meet, but it's kind of the back of the throat. We can't really see that on any models, but you should be familiar that you have one because it's important. So as this food gets masticated and chewed and mixed with saliva, it's going to come down another structure called the esophagus. The esophagus is the food tube, comes down. We'll talk about muscle and peristaltic activity. Peristalsis is the wave-like contractions of the smooth muscle that move a lot of this food through the digestive system. Very important. Some other structures that we have on here. As that food comes down through the esophagus, it will get dumped into the stomach. So the stomach that we could see right here, we'll dive into some details with it. Obviously, in lecture, we'll talk about hydrochloric acid and the mixture and what this stuff's called. But the stomach is very muscular and it has a lot of acid in it. It is also lined with a layer of mucus, and we will talk about the three different types of muscles that are in there, and the gates that control the entrance and exit to the stomach. These are called sphincters. The top one is the esophageal sphincter, and we'll talk about the other ones when I have pictures, and I'll show you the duodenal. Clearly, you need to have gates so this acid is not just leaking out everywhere. And after the food mixes in the stomach a little bit, it comes out, and you can't really see where it comes out, but it enters this massive tube work of small intestine. So you can see three structures that I will highlight: the duodenum, the jejunum, and the ileum. These three structures are the various regions of the small intestine, right? Um, the duodenum is right where it's coming out of that stomach. The jejunum's pointing to it here, but it's kind of that middle region. And the ileum is that bottom region. So I'll show you where on the models we could specifically see those and where I would ask you. Once that food exits the stomach, it gets bombarded with chemicals, lots of digestive enzymes, bile secretions, etc. And all of these things start to mix with the food in the small intestine. And the main goal for the small intestine is to break down all these nutrients and absorb them. This is our primary site of absorption. This is where, and we'll show you the villi and all the various anatomical structures that are very, very important for breaking down and absorbing these nutrients. It's a very long, and its anatomy specifically serves its function, right? Once this foodstuffs, good color here for you, hopefully gets broken down and absorbed. We want to maximize that for max nutrient absorption based off what we're putting into our body. Whatever remains gets dumped into the large intestine. Whatever passes through the large intestine and does not get absorbed or something happened to it like bacteria break it down, obviously it will come out of the anus and we call that feces, right? So food comes in, main goal, break it down, absorb what we can, whatever is left over gets eliminated. So the large intestine starts off with this little appendix-looking structure here. The first big part is called the cecum, and these are just generalized regions. Then it goes up the ascending colon. Then it will come across the transverse colon, down the descending colon, stored in the sigmoid colon, obviously out the rectum and the anus, respectively. This is the pathway food takes. There are some other accessory structures that we will discuss, and I will do these in, I don't know, reddish. And these accessory structures are the pancreas, which we've talked about before because we talked about it in the endocrine system. It makes very, very important hormones to regulate blood sugar. Now we're going to talk about it from a digestive perspective. The pancreas is tucked in behind the stomach there, and it is producing tons of digestive enzymes that are getting dumped into the small intestine. The liver, oh, so important. Tons of different roles as far as digestion goes. It produces bile. A lot of the food that you absorb gets shipped there to get kind of processed and filtered. It modifies different types of cholesterol, lots of different roles, stores glycogen, etc. And lastly, the gallbladder, which is that little green guy tucked in behind there. And the gallbladder primarily stores bile that was produced by the liver, right? Gallbladder stores and releases bile, and it also dumps it into that duodenum. Very, very, very important area of the small intestine is the duodenum. Lots happening there. But those notice food doesn't necessarily pass through them. They're accessory structures outside of the tubular network that is the digestive system. So hopefully that makes sense. We'll go through and we will highlight some of these structures. Obviously, you have real anatomy pictures, cadaver stuff you have to be familiar with. It looks a little bit weird. Obviously, some stuff's atrophied. There's no food in it. So it looks a little bit different and you should be familiar with it. Clearly, all the stuff that we talked about, you need to know. Check your list. Make sure you know where it is. And we talked about our glands, right? So here, and we have a model I will show you, but we have our parotid gland. We have our sublingual gland, and we have our submandibular gland. These glands can be located here for the parotid, sublinguals underneath the tongue, and the submandibulars behind the mandible. Obviously, we have a tongue. It's part of the digestive system as well. And obviously, we have an oral cavity or mouth. As we move on to this, it's just showing you again the tongue, sublingual, submandibular, and you cannot see the parotid gland on this model. It is going to be on the other side, which would be covered because we're only looking at the left. As we move down, this is the first picture, so I'll highlight what we can see here. The first thing I see is the liver. The liver is always going to be that darkish red structure. In addition, we can see the stomach over here off to the side. Once I take some other structures out, we'll be able to see it more clearly. On this picture, we can see obviously all of this is small intestine. I usually highlight the jejunum over here, the ileum down here, and the duodenum up here because it would be coming off the stomach. We can see, pick a color I haven't used, the cecum, the ascending colon, the transverse colon, the descending colon, and a piece of the sigmoid colon. So on this picture, you can see some things, but you cannot see everything. On this picture, we could see a lot more. So we're going to start off with the esophagus and the esophageal sphincter. We can see the stomach. And within the stomach, you can see those folds. These folds are called rugae. Right? That's what these arrows are pointing to. Rugae. And we will see that when I go into a little bit more detailed view of the stomach. We have that sphincter down there. Obviously, this part right here is the duodenum. We know how important it is, so we're going to put a star next to it. Then you would have your jejunum down here, and you would have your ileum over here. We can also see the pancreas in this picture, right? That's the little pancreatic duct that secretes all this stuff out here. We can also see the gallbladder, obviously the liver, and this duct where it dumps in down by the duodenum, it's called the common bile duct. That gallbladder is storing bile, travels down those little secretory ducts and gets released into the small intestine in the duodenum, which is important. Lastly, we can see the cecum, ascending colon, transverse colons cut off of this model, descending colon, and sigmoid colon. Be familiar with the picture, be able to label your stuff. And then this is kind of like looking at the top half. And then this picture is looking at the bottom half. So obviously at the top, we could still see pancreas, common bile duct, duodenum. But down here, we will highlight the various areas of the large intestine, appendix, cecum, ascending colon, transverse would be across here, but it's not, descending, sigmoid colon, rectum, and anus, respectively. You can also take note where this ileum dumps out into the large intestine, and our jejunum would be over here. Makes sense. Looking at the anatomy of the stomach, we can see the rugae, aka those folds. We can see the pyloric sphincter. We can see the esophageal sphincter. We can see the duodenum, all things we've talked about, but we're going to add in 3 new components. And those 3 new components are muscles. We have longitudinal, circular, and oblique. The longitudinal ones run long ways. The circular ones run in a circle around. And the oblique ones run kind of at an angle. So we have a model, and I'll obviously show you those. You can call them layers or muscles or whatever you want. Longitudinal layer is the simplest thing to do. Circular layer and oblique layer, respectively. So on here, looking at the model of the stomach, we can see the gallbladder. We can see that common bile duct dumping bile into the duodenum. Clearly the duodenum. We can see the rugae, those folds in the stomach. We can see the sphincters. And muscle-wise, we can see the longitudinal, circular, and oblique layers. Longitudinal, circular, and oblique layers. We have a liver and pancreas, which we discussed as accessory structures, as well as the gallbladder. So you don't have to stress too much about this. Just know the anatomy for lab. But you can see that gallbladder. Obviously, the liver's making bile and it's stored in that gallbladder, and then it will get dumped out. We can also see that pancreas is right there, also dumping out stuff into the duodenum, aka this area right here is extraordinarily important. In lecture, we will discuss all the things that are produced and released, what the significance of bile is and what its role is, as far as emulsifying fats, etc. And that is the picture. This is a picture of the liver. We will talk about various blood vessels that are extremely important, like the hepatic portal vein and things of that nature. But for now, that's the picture of your liver. And this is your hepatic portal vein, a very, very important group of veins. Now, this is showing you a villi. If I looked at a cross section of the small intestine, there would be these little folds. And these little folds have little finger-like projections on them. I should say, this is the finger-like projection. And then there's little hair-like projections. These are little hair-like projections. The point of these is to increase the surface area of the small intestine so we can absorb as much as humanly possible. That's the goal. More little folds, more little fingers, more absorptive area. We want to maximize nutrient absorption. So this is our villus. And then on here, you can see we have these little microvilli, all these little hair-like projections. You can also see that there's groups of blood vessels inside here. So this is a villi. The little things are microvilli, and we're going to zoom in on a microvilli, right? This is one of those little hair-like projections, and we can see that there are some structures that I want you to know. Number one, this picture is a microvilli. Number two, we have red blood vessels in here. The red ones are called arteries. The blue ones, which we could see running up over here, are called veins. But because they're small, we call them arterioles and venules. The arterioles deliver blood to this little finger-like projection, and the venules will take the blood away. Hopefully, everything we needed got absorbed through this epithelium in the little microvilli, and enters the blood. It enters the blood through these capillaries. Some stuff, fats specifically, will travel through the green structure. And the green structure is called a lacteal. So we should be familiar with the structures that make up these little microvilli and the blood vessels found within it. The only thing I did not mention, I mentioned it, but I didn't label, is the epithelium. There is a specific type of epithelium that we will discuss that lines the inside of, in this case, the small intestine, that allows for specific absorption and transport of nutrients through that epithelial wall. Extremely important. But for that, epithelium. For now, perfectly fine. Obviously, you have a key like we did on the last one to see all models and what's labeled, but I pretty much went through it. And you could obviously rewatch it, take pictures, whatever. And now we're going to do a little bit of digestive histology. The first thing that we are looking at is in the tongue. This is called a papilla. And what you can see on here are the taste buds on the side. What a papilla is are the bumps on your tongue. And then within those little recessions, you have taste buds on the lateral walls that allow you to taste various types of flavors, etc. This is a zoomed in version of one of those taste buds, and we can see what it looks like. There's various components to it, to supporting cells and various types of, neuroepithelial cells. This is the little taste pore where the tasting happens, but recognize this slide as a taste bud. And now we're going to go through different types of digestive tissue. We have three major ones that I want you to focus on, but we'll have four on the list. The first one is called the mucosa. The second one is called the submucosa. Then we have the muscularis, and the serosa is the outermost kind of connective tissue that separates it from everything else. So the mucosa, submucosa, and muscularis are from the inside outwards, right? The mucosa is the inner layer, the submucosa is the middle, and the muscularis is the muscular layer that does that peristalsis. And the serosa is the outside that connects it or prevents it from connecting to everything else and keeps it as its own organ. So on here, we would recognize that. Number one, this is the esophagus. Number two, we can see that we have this mucosa, always going to be the inner layer. We have this submucosa. Maybe I'll highlight it for you in specific colors, make it easier. Try to make it easier. And then I'll do that. Submucosa. Muscularis. And adventia is also serosa. And that'd be the outer. On here, we can see the stomach. We have a much larger mucosa, right? We can see the submucosa, and we can see the muscularis. Obviously, over there would be the serosa. Stomach, thick layer of mucus, super important, prevents that acid from burning the stomach itself, as well as some other functions. You can see these folds I was referencing on this picture. We could see the three different areas, the duodenum, jejunum, and ileum of the small intestine. These circular folds, again, are the entire small intestine. Most of it is covered by those hair-like projections and villi. These are what they look like, right? These are part of the mucosa. Underneath that would be our submucosas, usually going to be lighter in color. Obviously, our muscularis and serosa, respectively. These you can see mucosa, submucosa, muscularis, and a little bit of that serosa. But recognize this slide as the duodenum. Right, we could see like pancreatic duct and things coming in. Recognize this one as the jejunum. You can see that we have lots of little projections. Tons of villi, still the same four layers, but this is the jejunum. I remember it because you could see that little fold there. This is the ileum, right? So we're going duodenum, jejunum, ileum. This is just showing you one of the villi. In there, we have some goblet cells which secrete mucus throughout the digestive system, but it's just highlighted here. Ileum. These are goblet cells, right? They secrete mucus. You just saw that. This is a picture of the large intestine or the colon. And we'll have a few on the liver. We'll talk about it a lot in lecture, but the liver is mainly made of two structures that I'm going to discuss. It's made of a lot of stuff. The first structure I want to discuss, or cells, I should say, are called hepatocytes. Hepatocytes are liver cells. They are very busy doing lots of things from filtering to detoxifying to converting things into other things. So the hepatocyte is the cells of the liver, right? They form structures. You can see how they kind of form these lines, if you will, right? These are called cords. The spaces where the blood is coming through to be filtered have two different names. One, the big tube that brings it in is called the central vein. We can see that here. And then you have all these little sinusoids. Let's see if I could find you a good. These are all sinusoids over here, even though I'm drawing in blue, so you probably can't see. And we can see the label there, right? So we have the hepatocytes, which form cords, and we have the sinusoids, which are the open spaces where blood's going to be going through. You can see the same picture here, right? Lovely. And this is the liver. So we can see the central vein. And the thing is, the cells, you can call them hepatocytes, but they form cords, are all the darker things. The sinusoids will be the spaces. So I think the next one, if we have it on here, would be a little bit better. So recognize hepatocytes versus sinusoids. We could see this real good here. The darker cells are the hepatocytes, and the sinusoids are the lighter sections. And you can see that there's red blood cells in there and a bunch of other things. We've seen the pancreas before when we did the endocrine system. We know that the darker areas are pancreatic acini or acinar cells. And we should also know that the lighter section are the pancreatic islets or the islets of Langerhans. Previously, we've spent a lot of time talking about the islets. In this situation, it's more about the acinar cells. Those are the cells making digestive enzymes. And we're talking about digestion. This is just zooming in a little bit, so you can still see those acinar cells, which are purple, and they have purplish pinkish, and they have all these little digestive enzymes and things in them that will be secreted out into the small intestine. That's your anatomy. Talked about all the structures, we labeled every picture, and we did a little bit of histology. Now I'm going to wrap this up by talking about some macronutrients, what they are, what they look like, and then we'll talk about some enzymes at the end, and we will be on our way. So we have three basic macronutrients that all food is made of: carbs, fats, and proteins. Most food things are made of a variety and mixture of these three macronutrients. When we break down carbohydrates specifically, we have simple and complex carbohydrates. I'll show you what they look like, but complex carbohydrates have way more sugars attached to them, right? We talk about things like, bread, legumes, fiber, tons of stuff like that. And our complex carbohydrates look like this. Bigger. Obviously, this is only showing 4 sugars. So you should be able to recognize a sugar molecule, a disaccharide, and obviously a polysaccharide. That six-sided figure is glucose. So depending on how many of these are connected together, it could be a monosaccharide, disaccharide, or polysaccharide. These are your examples of polysaccharides. We talk about glycogen all the time, cellulose in reference to fiber, and obviously starch, which is food that we would consume, which is a polysaccharide created by a plant. But you could just see big chains of sugar. Down here, we have a picture of a protein. Protein is a chain of amino acids. Obviously, we'll show pictures, but your meats and things like that. And this is one amino acid, and it's showing you how they are connected together. You don't have to understand the biochemistry, but know that proteins are made of amino acids, an amine, and an acid group. Clearly here, our proteins are our meats, dairy, eggs, legumes, which we see right here, fish, you name it. Proteins are made of amino acids. They're extremely important to the diet. We have to consume a certain amount of protein so we can build and repair tissues so we don't develop serious diseases. We have fats. I will show you what these fats are. This is a triglyceride. We can recognize that because it has a glycerol backbone and 3 fatty acid chains. This is a cholesterol molecule. We talk a lot about cholesterol as far as maintaining low bad cholesterol and having high good cholesterol and a variety of different things. The liver is the one that's processing this, and in lecture we'll go through into some detail. This is a free fatty acid, which is basically just one of those. And we have triglyceride, which is just another way to look at the same picture. So these are the same. Obviously, our fat sources include, butter, oils. Everybody thinks avocado. There's different types of fat. There's saturated fat, unsaturated fats. All these things will be discussed. And here's what they look like. Now, in order for something to be digested, almost always, we're going to have an enzyme. Enzymes are extremely important. Talk about them a lot more in lecture, but I want to bring them up here. An enzyme is a protein that is going to, in the case of a digestive enzyme, recognize a substrate and break it down. So maybe it recognizes a protein and it chops the protein up. Maybe it recognizes a starch and it chops the starch up. And I'll give you a list of enzymes that we will be discussing. But as far as terminology goes, a substrate is the thing that is going to get broken down. Or in this situation, the thing that is getting built, but we're talking about digestion, so I want you to think about breaking. The enzymes live in that duodenum, and they just go around chomping up all your food products and making them very, very small, so then they can be absorbed through those villi. This is a list of enzymes. You should be familiar with it for both lab and lecture. You can see that a lot of them end in ACE, and this is a practice sheet for you. So you could print it out, you could do whatever you want. Where does the enzyme come from? Substrate is what does it work on? And the end products are what does it break it into, right? So maybe you name it, lactase breaks down lactose sugar, which is a disaccharide, into its 2 respective monosaccharide components, right? Amylase, going to break down complex carbohydrates or sugars into glucose. All the pepsin, peptidases, etc., trypsin break down proteins. You got it. We have a few hormones that are involved specifically in the digestive system. You can be familiar. Again, we talk about this more with lecture. As far as GIP, right? Gastric inhibitory peptide, CCK, ghrelin, things like that. They all play a specific role. As far as triggering stomach activity, satiety, hunger, etc. And you can fill this out and be familiar with that. I may ask you maybe one question or two on it. And this is just showing you the process that I've described multiple times about how things get absorbed, right? We have to break things into glucose, then it gets absorbed. We have to break things into amino acids, then they get absorbed, right? Dipeptides are just two amino acids, et cetera, et cetera. Um, and fats. So this is just showing you the bottom picture, the capillaries we discussed, the lacteal, the arterioles, and the venules, and how some of these are being transported in. The sugars and the amino acids will go into the blood, and the fats will go into the green one, aka the lacteal, the lymph, right? This gets shipped to the liver for processing. The lymph obviously gets recirculated back into venous circulation and then will eventually be processed. Just zoomed in version of the same thing. Could ask you questions on this picture. Should be familiar with it. And then you have a bunch of real anatomy. You have it. You have your homework. You'll do it. I don't need to go through this with you, but everything in here is labeled. Could ask you those pictures. Be familiar with it, right? Pictures of the small intestine, the different regions, duodenum, jejunum, and ileum, respectively. We could see the rugae, all types of things. And hopefully that makes sense. What did we talk about? We did the basic anatomy of the digestive system. You should know all your structures, have a general idea of what they do because that will correlate directly to lecture. We did some histology, obviously labeled our models. And then we talked a little bit of basics about macronutrients, carbs, proteins, and fat. And that's the gist. So you completed this lab, and I will see you for the last one in the next recording.